common traits with genetic susceptibility. And our first speaker will be Dr. Doumatey, who's going to give us a presentation on the effect of obesity on transcriptomic changes. Just before she comes up for her presentation, I'd like to make a quick announcement, and that is at the end of the day today at about 5 o'clock, we have a strategic meeting which everybody is invited to participate in. But just to make the facilitation of that section go smoothly, we seriously encourage you to develop questions that we can use to make the section go smoothly. And the purpose of this strategic planning meeting is really for us to brainstorm together as a group, to look at the challenges and opportunities that we have experienced over the years in terms of genomics and other biomedical research on the African continent, and to see how we can move this by coming together to look at what are the success stories, the challenges, in a way that we can stimulate discussions for the things that we want to do in the next coming years. So please think very carefully about this and write your questions down, and you can give them to me, Michelle, or Sonia, and at any point, preferably before lunch, today. So thank you very much, and I will invite Ayo to give her talk.
Good morning. I changed the title of my talk a little bit so I can cover more of the molecular dogma and use the tool applied to obesity. So the talk will be covering changes in DNA that has been associated with obesity, as well as changes at the transcriptional level and also at the protein level. So that all of us start with a basic definition of obesity, is just an excessive accumulation of body fat that can be either visceral or all over the body. And the most commonly used variable to define obesity, which is actually easier when you're doing an epidemiology study, is BMI, body mass index. So the reason we use those measures is because it's easier to be measured when you have a large population, and also it has been shown to be associated with more complicated methods such as DEXA or BIA. Somebody will be defined as obese if the BMI is greater or equal to 30 kilograms per meter square. So obesity have different subtypes. Depending on where your fat is accumulated, actually, there will be implication for pathogenicity. So an apple-shaped obesity usually is obesity that is all around your body and especially around the waist. So when you have that type of obesity, which is usually seen in men and also in Asian population, you tend to have a lot of comorbidity, hypertension, type 2 diabetes, and also a lot of other non-metabolic disorder. Whereas if you have a pear-shaped obesity, which is seen a lot in women because the fat is accumulated around the hips, that obesity is actually less pathogenic. And these individuals tend to be healthier despite a high body mass index. And I will come back to that group a little bit more later in the talk. Because this group have been now referred to as the metabolically healthy obese individual, and it's a subgroup that is really important to understand, because even though some of them have a BMI of 50, they do not have any of the comorbidity, and they carry that weight for a long time in their life before they develop any comorbidity. So we all know now that obesity is a global epidemic. In 1985, you can see the map of the world where, for instance, Africa only most of the countries in Africa have less than 2% obesity rate. The exception here would be Northern Africa and South Africa, where they actually mirror Western countries. And on the right side, in 2014, you can see that in most countries in the world, the obesity rate now is around 30%. And actually in the US, it's greater than 30%. And again, the same pattern is seen in Northern Africa and South Africa, where you can see that the color are almost like in Western countries, especially in US. So when we zoom in a study that we conducted in West Africa and East Africa, you can see that the trend is the same. The first point on the graph is actually data that was collected in Nigeria and Ghana between 2001 and 2006. And you can see that the average BMI was around 25 for the control, and around 26 when you have the overall population, which includes diabetic and control. And the last on the top is actually looking at all the population together. And you can see that as we go on in the study, which on the second point, the average went up of about 2 units, and that was conducted between 2006 and 2014, roughly. And now around 2016, we have a huge increase in BMI, and almost everybody in the study is around a little bit below 30%. And at that point, you have 3 countries there, Ghana, Nigeria, and Kenya. And you can see that the trend is continuing even when you go at country level. So obesity is one interesting disease because it affects almost every organ system in the body. And as shown on this picture, you can see that it affects anywhere from the brain to the bone system, and which is the challenge with this disease. When we talk, we usually talk more about the metabolic effects, but there are a lot of other things that are coming with obesity, which is a challenge, and it costs a lot more actually to maintain these individuals that become obese. So when we talk about obesity, I like to refer to the macro changes, which I just showed on the other slide, but also you have changes at the molecular level, and it can go anywhere from DNA to protein. And what I'm going to do in the next couple of slides is to show changes that are at the DNA level, RNA level, and also at the protein level. So this picture is just depicting all the genes that has been associated with any anthropometric trait in human population. But I want to emphasize that in this picture, which I do not expect you to read everything in there, it's about roughly 120 loci that has been associated with obesity. And most of these genes has been found in Caucasian population. Few of these genes here has been replicated in African population, but the majority are in Caucasian. So what we've done in our lab is to kind of focus in African population and see if there are other genes that may be explaining obesity, common obesity in African population. So in this study, what we basically did is to use our cohort in West Africa and East Africa to look at other variants that may be explaining obesity. And we found, here you have the map of the country that have been included in the study. The discovery phase included about 1,000, roughly 50,000 individuals, and we validate the finding in also an independent cohort of West African, and then replicate the finding in West African and African American. So I'm showing here the Manhattan plot. We found one SNPs is a rare SNP with a frequency of less than 1%, and it's only found in the African descent population. So if you were to do this study in European, you will never pick up this variant because it's not polymorphic in our European population. And as you can see on the right side, the C allele is actually associated with a weight gain. So most of the carriers of the C allele have a BMI of around 31. And then the T allele, which is the most common, have a BMI around 26. They're overweight, but they're not obese. And then when we look at, we wanted to see if this variant actually have any effect or any association with the level of protein of the gene in which the variant is found. And you can see that the C variant actually is associated with a higher level of SEMA4D in plasma, telling us that there may be something going on between the variant and the phenotype. So in here, what I'm showing is that you can pick up a variant and also do a genotype-phenotype association and found that your variant may have an effect on the expressed protein. Here, again, I want to emphasize that this is is not a longitudinal study, so we cannot infer causality. All we can say is it's correlated. To infer any causality, you may have to do a longitudinal study. So next, I will talk about gene expression changes in obese individuals. And what you want to know here is to see if at the expression level, you can see any change that is associated with change in BMI or any other obesity trait. This particular study has been conducted using an adipose tissue, which is more specific than using whole blood. But the challenge with that is you have to go, it's more invasive, and you may not get approval to do a lot of individuals. Most people would not agree to have their fat taken. But most time, it's easier to have this sample taken when people are going through bariatric surgery. Because anyway, they will be taking the fat out, and you can request the fat to do your gene expression work. So this paper has been published a couple of years ago, and is a particular study because what we wanted to see is to see the effect of obesity on diabetes. So this individual, both groups are obese, but one group is diabetic and the other is not. And again, the tissue was the omental adipose tissue, which is taken deeper from the tummy area. And we use a microarray that covers almost the whole genome. And Dr. Kasim talked about the method yesterday, so I will kind of assume that everybody here was at that talk yesterday and will not talk too much about the methodology. So this is the characteristic of the individual, and the mean age is 41 in both groups. BMI, as you can see here, these are morbidly obese individuals greater than 53. And then for the diabetic individual, the glucose is kind of well controlled, is around 128. So you can see the Venn diagram here, and clearly what you can see on the right side is the obese without diabetic, and you can see that the profiles are different. So there is definitely a change in the gene expression of this individual. And what you can do here on the right side, you have the list of genes that we pick up. Just looking at the gene name, it may not tell you much about the function of this gene. So you can use a bioinformatic tool to put those genes in a pathway. So in this particular case, these are just again the list of the genes that we pick up. And when you do the pathway analysis, one thing that comes up is the telomere extension. And again, remember this study has been conducted in adipose tissue, and these individuals, some of them are diabetic. And you see that the first pathway is actually shortening of telomere, which has been associated with diabetes without obesity before in other studies. And this study confirmed two things. The same pathway that you may pick up in a whole blood can also be picked up in specific tissue that are associated with your phenotype. So here, the question you're answering here, now that I have those different expressions, do I know what is driving the difference in expression? And again, this is a bioinformatic tool that you can use called upstream regulator analysis. And in this particular study where we take the 25 genes that are differentially expressed, interestingly, all these genes are being regulated by microRNA. And what we found is that 2 of the microRNA have never been associated with either obesity or type 2 diabetes. So what you can do from here is to actually knock down this microRNA and see the change on the tissue or on the whole animal. And here, now I'm going to focus on the subgroup that I was talking about, the metabolic healthy obese individual. These individuals are morbidly obese. And as you can see here, this chart was a little bit older. At that point, the prevalence of metabolically healthy obese individuals was around 10%. But this was using a very strict definition. If you kind of, because most people at this weight will have at least one comorbidity, which is usually hypertension. But in this definition, they use 0 comorbidity, which explains why the prevalence of this subtype is so low. But if you just relax the definition, including one comorbidity at least, what happens is that this prevalence quickly goes to around 40%. And that's the definition that has been used now. So these individuals, they have no blood pressure, no insulin resistance, no type 2 diabetic or pre-diabetic, and they have low inflammation. And understanding this individual will help actually find what can be wrong with individuals that are developing comorbidity. So it's a good way to actually find new drug targets if biomarkers are able to discriminate this individual. So here what we did is to look at the protein level and screen all the protein in serum. And the reason being that you want to find biomarkers that will tell you, and specifically when you have a cohort of obese individuals, that you can classify them. And importantly, this group of individuals that do not respond to standard care. If you put the metabolic healthy individual on a standard diet, what happens to them is they actually worsen. They develop insulin resistance. So in clinic, it's actually good to know which treatment you should put this individual on. So again, what we've done here is to just a shotgun proteomic study. And what we found is that, I will quickly go through the demographic, but what you want to see here is that those 2 groups are really different based on the comorbidity, because that's the definition that was used. And one key that I want to you guys to focus on here is that these individuals actually are really well characterized by their lipid profile and inflammation. So if anything you want to take out of this stuff is that if you just have a panel of inflammatory markers and lipids, you can do a good job actually discriminating this individual from the others. So this is small to read, but these are the proteins that we found that were differentially expressed between the 2 groups. And when you classify them, again, they really come down to inflammation protein and also lipid protein. And most of them are in the group of the ApoB100 protein and for the inflammation, they're in the acute phase resistant protein classes. So here, when I just use 10 of the top protein that we found, you can see that you can discriminate the two groups. The red are the metabolically healthy individuals, and the blue are the unhealthy. So just using 10 of the protein, you can actually discriminate them. And here, using again pathway analysis and throwing all the proteins that are differentially expressed, you see that in the red rectangle, all the pathway are protein, inflammation pathway and lipid pathway, telling us that inflammation and lipid are key in developing comorbidity. So if you can even control those two factors by any medication, you may extend development of comorbidity in this obese individual. And as I alluded to before, microRNA tend to be now, we have seen that microRNA is actually regulating a lot of the changes that we see. So even using microRNA as a biomarker would be also another way of discriminating this individual if you have access to microRNA sequencing or typing. And in conclusion, it's just that obesity is associated with number of change at every level of the molecular dogma, from DNA to protein, and the easiest in clinic to use would be protein. So if you can use some of those markers that we found in your clinic, that would be a good start. And digging more into those pathways may be target for new drugs to control development of comorbidity. And while we're doing all this research, I would want everybody to just keep exercising, because we do know that we see changes with exercise at the transcriptional level. And I would like to acknowledge all my colleagues that are in the audience, and that helped me do all this work. Thank you.
Questions for Ayo, please? Yes, Michelle?
Thank you so much, Ayo. That was a fantastic talk. I just have to ask you, we always say that obesity is very different in men and women.
Yes.
Have you done a sex stratification for your expression studies and also your protein studies?
When you do the stratification, in general, the expression differential, when I talk just about the expression level, at the RNA level, you tend to find the same gene, but there are a couple of genes that are specific to women, which we know that there is a sex specific gene when you talk about obesity. So you see the same thing with gene expression. But for the protein, I didn't see any difference when I did the proteomic analysis. But the reason is because all the individuals included in the proteomic study were all women, because I used a very stringent definition, and when you do that, the prevalence of healthy obese is really high in women. So you end up having just women in your data. So I think next thing we have to do is to try to get some metabolically healthy men, which is really rare, because many tend to accumulate their fat around the tummy. So yeah.
Just one more question, please, and that so that we'll be on time, okay?
What's the cause of the global increasing trend in obesity?
What is the cause?
The cause of the global trend of increase in obesity.
The short question is change in diet and lifestyle. That's the short answer.
Okay.